Hello, I am Dr. Pokraj Guho, Assistant Professor, Department of Zoology, Gorbeda College. Welcome back to my course Basics of the Immune System, and today I will deliver lecture on core 4 of my course, that is autoimmunity. Okay, so about immunity, the first thing is I want to mention is that the disorders of the immune system. You can see that immunodeficiency is very little found in the population. Hypersensitivity is found in large number. A large number of people are hypersensitive to different antigens, and we can find autoimmunity. In most of the cases, it is misdirected. Means in some cases undiagnosed. In some cases, we can see that the people are less careful about it, are less concerned about it. So, horror autotoxicus. Many of us who study immunology have heard the term. Literally, it means the horror of self-toxicity. Means anything that is our self when it becomes toxic to us. The fear is known as horror autotoxicus, a term coined by the German immunologist Paul Ehrlich, 1854 to 1915, to describe the body's innate aversions to immunological self-destructions. Autoimmunity is the failure of an organism in recognizing its own constituent part as non-self, which allows an immune response against its own cells and tissues. Any disease that results from such an aberrant immune response is termed as autoimmune disease. Means when the body cannot recognize its own antigens, then only it attacks its own tissues and cells. Any disease that results from such aberrant immune responses is termed autoimmune disease, and autoimmunity is often caused by lack of germ development of a target body, and as such the immune response acts against its own cells and tissues. So you can see there is no response to self, and non-self or foreign we have strong response. So before going into the details of autoimmunity, I would like to say something about the general tolerance mechanism. We can see there is a lymphoid precursor, and from that maturation of clones not specific for self antigens present in primary lymphoid organs, destruction of lymphocytes specific for self antigens present in the generative organ, maturation of clones that suppresses self antigen recognition. So we can see what is tolerance. Tolerance is the capacity of the body to recognize self antigens and not to trigger immune response against it. So we can see that from the lymphoid precursor, when differentiation and development takes place, so maturation of clones occur which are not specific for self antigen, means they are specific for foreign antigens, and any clone that is specific to self antigen are destroyed. In the middle, all the cross marks shows that the destruction of lymphocytes specific for self antigens present in generative organs. The failure of tolerance gives rise to autoimmunity. So whenever the clones are formed against the self antigens, when the immune cells, the clones of the immune cells are formed against the self antigen, then only you can see that tolerance is broken down and autoimmunity is crossed. Autoimmunity occurs. Failure of tolerance to protect host from self-reacting lymphocytes, destruction of cell proteins, cells and organs by autoantibodies or self-reactive T cells. 3% to 8% of individuals in the industrialized world suffer from autoimmunity. Now, genes, in one hand, the genes, the environment, and immune regulation, all these three things together are the causative factors behind autoimmune diseases. What are the mechanism? The mechanism involved failure to detect autoreactive lymphocytes, central tolerance failure, peripheral tolerance failure, molecular mimicry, we have heard of this term and we will explain it, abnormal presentation of self antigen, aberrant expression of major histocompatibility complex, Cas2 molecules, coupling of self and non-self antigens, overproduction of self antigen, disclosure of cryptic T cell epitopes, release of sequestered self antigens, epitope spreading, polyclonal lymphocyte activation. Altogether, autoimmunity may be due to immunological, genetic, and viral, drug-induced, etc. Most insistent disease by multiple mechanism, difficulty in diagnosis. So we have already gone through this part, that it is genetic factors, infection, environmental exposure, both causes the immune regulation to disrupt, and it results in autoimmunity. So mechanism is antigenic alteration. The antigens are altered. We cannot recognize sequestered antigens, those antigens which are not exposed to developmental processes, and then they are exposed to any part of the lifetime and can cause autoimmune disorders. Cross-reacting foreign antigen, molecular mimicry, polyclonal activation of B cells, forbidden clones, and at last alter T or B cell function. Antigenic alteration, physical irradiation, photosensitivity, cold, means here the antigens are present in the body are altered, are damaged, which are then recognized by the immune system. Viral infection can cause like something similar to antigenic alteration. A sequestered antigen, as I have already told, self-antibody present in closed system, not accessible to immune system, lens protein like sperm, etc. Immunological tolerance not established during fetal life, means they are not exposed to the developmental system, these antigens, and again when they are exposed they causes immune reactions to occur. You can see sperm antigen in the puberty, it causes the autoimmune disorder. Cross-reacting foreign antigen, similarity between foreign and self antigen results in type of autoimmune reactions to self antigens. Identical peptide sequences in epitopes of microorganisms and self antigen. HLA-B27 arthrodogenic strain of S. flexneri both have the similar peptide sequence, which can result in molecular mimicry and autoimmune disorders relating to HLA-B27. Rheumatic fever is a classic example of molecular mimicry. The forbidden clones, breakdown of immunological homeostasis leads to cessation of tolerance and emergence of forbidden clones of immunocompetent cells. Immune responses against self antigens occur. Injection of self antigen with fluids adjuvant can also result in autoimmune disorders. Alter T and B cell function, enhance the helper T cell, suppressor T cell function. The loss of suppressor T cell function, because suppressor T cells is the one which plays an important role in suppressing autoimmunity. But we can see if these cells loses their function or becomes less functional, then autoimmunity can occur. Defect in thymus, stem cell development, macrophage function, etiotypic and antireotypic network defect. The organ-specific autoimmune diseases and systemic autoimmune diseases are there. Organ-specific include type 1 diabetes, Gout-Pasteur syndrome, etc. Systemic autoimmune diseases involve rheumatoid arthritis, scleroderma, etc. So before going to the examples, I would like to more about the examples I will give in the material. And now let's go for some tool discussion.
Sir, do autoimmunity occurs in the endocrine system?
Yes, of course. Crohn's disease, celiac disease, ulcerative colitis are examples of endocrine autoimmune diseases.
Thank you, sir.
Thank you for your patient hearing. Visit me at pokrajclasses2022.genomeo.com or call me at 977 599 5422.